Hello and welcome to another lecture. This lecture is about the plant hormone auxin. So before we talk about the plant hormone auxin specifically, I want to spend some time going over some of the hormones that we've already discussed and pointing out some of the other important hormones that we will discuss later on. So just in general, plant hormones have a lot of different responses. And it's almost impossible to just remember all of the responses for each of the hormones. But there are some main themes of these hormones that would be good to remember. Auxins, which we will discuss in great length today, are mostly responsible for cell elongation, apical dominance, so it means that the top of the plant will always grow and repress the outgrowth of the side shoots, and directional growth, so tropisms, gravitropism, phototropism. And we'll come back to that because we discussed this already in the previous lecture. Cytokinins are another important plant hormone, and they are mostly responsible for cell division. They have other functions as well, but they're best known for their role in cell division. Gibberellins is a class of hormones. There's lots of different variations of those, but together as a family they're called gibberellins, and they are important for germination and for stem elongation, as opposed to cell elongation, individual cell elongation. So this is lots of cells, cell division, cell elongation that will allow the stem to elongate. And as you can already see, these hormones also interact with each other to fulfill all these functions. Another important hormone is called ethylene. So that's a gaseous compound. That is the ripening hormone. So that's responsible for ripening a set of species, the fruit of a set of species. They're also known as climacteric fruit. We'll come back to that later on. We don't need to know that right now. But ethylene is important for ripening and also for growth inhibition. So this is an inhibitory hormone as well as a ripening hormone. And then there's abscisic acid. We've discussed this already. That's why it's in red. Briefly, we just talked about its role in stress avoidance from drought, closing of the stomata. So that was part of the last lecture. And then there's brasinosteroids. Brasinosteroids are very similar to steroids that we find in animals, but they were first found in the Brassicaceae. Those are the mustard plants, and so that's why they're called brasinosteroids. They are also involved in cell growth, in development, early development of plants, photomorphogenesis. So those are the things that are working together with phytochromes to turn a seedling into an adult plant. And then there's jasmonic acid, and we've talked about jasmonic acid as well. Those are involved, the jasmonic acid is involved in activation of plant defenses. So today's topic is going to be auxins. Let me show you the structure of auxins. So this one, there's a number of different types of auxins, but the most common natural auxin is indole 3-acetic acid. So there's an indole ring here, and then on the 3 carbon there's an acetic acid. So that is indole 3-acetic acid. That's the most common natural auxin. Okay, so I said that auxin is involved in tropisms, in growth directional to gravity or directional to light. So this is an example of a plant that was a little while ago turned on its side from its pot that was growing straight up, and now it's sitting on its side. And you see that after a while, the plant has started to grow upright again. So this is called gravitropism. It's the response to gravity. Now the question is, how is this response mediated at the cellular level? That's what we want to spend some time on today. Why is it that the plant is growing up? How is it that the plant is growing up? What does auxin, that is mostly produced up here in the apical portion, in the apical meristem of the plant, How is that able to lead to this gravitropic response? All right, so we'll start on the inside of the cell. We're looking at some transporters. So we've talked a lot about transporters in this class before. There are two transporters that I want to spend some time on today. One of them is called OX1 and the other one's called PIN1. So these are the proteins right here and right here. And that is this green protein here and these red proteins down here. And as you can see, they're localized to the membrane of the plant cell. Here's a cell file of a cell that's on the top of this cell, which is on the top of this cell. So you can imagine this being the stem. It says here apex and base. So from the top to the bottom of the plant, cells are stacked one on top of the other. And in the top part of these cells, in the membrane of the top part of these cells, there's the OX1 protein, which is a H+ cotransporter. And on the bottom is the PIN1 protein, which is an efflux carrier. So I write here passive uptake as IAAH or active uptake via the transporter OX1. So when I say passive uptake, so IAA as the acidic form can diffuse, but very slowly into the cell. So this just goes in by its concentration gradient, will go in, but only in the acidic form. In the anionic form, it can be transported rapidly through this cotransporter. Once it's in the cell, it becomes deprotonated, as you see here. And as the anionic form, it gets transported out of the cell, where it then gets protonated again by the higher concentration of hydrogen ions, protons, in the cell wall, in the apoplast. So this gradient is maintained by one of the ATPases that we've already discussed in great detail with respect to many of its other functions. So the ATPase maintains the pH gradient, and that drives the OX1 uptake. And then the efflux goes through this carrier out into the bottom part, to the basal direction. So the whole reason for spending some time on this is to bring home the point that auxin is transported in a polar fashion. It's called the polar auxin transport model. So it's transported from the top to the bottom. That means there's a higher concentration of auxin towards the top of the plant and a lower concentration towards the bottom of the plant. And that, as we will see in a little bit, is part of the reason why the responses come from the apex of the plant and are slowly distributed towards the lower part of the plant. I'll come back to that in a second. Let me show you a way how this was studied. So we want to see how we know that the OX1 protein is found here, and why we know that the PIN1 protein is found here, using mutant analysis, of course, but also using some new techniques that we haven't discussed in this class so far. So let's start with this technique of the GFP fusion reporter constructs that you can use to track RNA expression. Last time I talked a little bit about certain RNA expression techniques. We talked about northern blot analysis, for example. We also talked about using different promoters and overexpressing a gene. So today what I want to show you here is that you can take a native gene, so that's just the gene X with its own promoter X, and you can swap its promoter and its reading frame with either a different promoter or a different reading frame. Last time we swapped out the promoter and gave it a very strong constitutive promoter. This time we're going to swap the gene out and we're going to put in what's called a reporter gene. So for example, there are two kinds of reporter genes that are commonly used in plant molecular research. One is called the GFP reporter construct, and the other one is called the GUS reporter construct. This one is a little bit more classic, if you will. It has some advantages. It's still used. The GFP reporter construct is a little more modern and is a little easier to use, but there are some disadvantages as well. So what you do if you make one of these reporter genes is you isolate the gene by PCR, for example, or you could cut it out of a vector by using restriction enzymes. And then you fuse, this is all happening in Eppendorf tubes, of course. So then you fuse using ligase enzyme the gene that you want to splice post the promoter of your choice. You put those two together in the tube, you add the ligase, and you let them fuse together here. So now this gene, this GFP reporter gene, is turned on whenever promoter X is turned on. So remember that gene X, of course, is only turned on when it's needed, right? So any kind of gene, whatever the gene is, has a certain promoter, and that promoter will determine when and where in the organism the gene is expressed. And the way that that works is by transcription factors that bind to the promoter. And when the right transcription factors are bound to the promoter, then that turns on the gene. Well, in our reporters, we have the same set of transcription factors that were originally controlling the expression of gene X, except now they are controlling the expression of the GFP gene. So that means that we can tell when gene X would have normally been expressed in the cell at the same time at normal times, except now we see a glow, a green glow. The GFP gene is now producing the GFP protein, and that is something we can visually see. And of course, we couldn't previously visually see where gene X was expressed just by looking at the plant. We did talk about one other type of experiment, of method, where you can sort of see that. That was the in situ hybridization assay. But there you have to first make sections, and you have to make a probe, and then you have to add the probe to the RNA so that it binds, and that way you could visualize the expression. In this case, you don't need to injure the plant. You just take the plant and you shine a blue light at it, like a UV light that will excite the green fluorescent protein, and then you can see where it's expressed. Or you can make sections if you want to look at a greater magnification of it. But it's a very straightforward, relatively straightforward experiment in order to determine where a gene is expressed. The Gus reporter construct is the same principle. You put the promoter in front of the Gus gene, and the Gus gene then will produce a protein, the Gus protein. For that, you can't just shine a light at it. You have to take the leaf, for example, or whatever tissue you're interested in, and bathe it in a substrate. And that substrate, together with the Gus enzyme, will then turn blue. Let me show you some examples of how this works. So the Gus reporter, here's an example for Gus reporter constructs, is usually done by using three types of experiments together so that you know what the experiment means by using two controls. The first control is an overexpression control. So here you use a strong constitutive promoter. You overexpress that Gus reporter. Of course, now everything will turn blue, right? So if this is a constitutive reporter, a constitutive promoter that's expressed at any time, anywhere in the plant, it will turn everything blue by using this Gus reporter gene. You will then take, for example, buds, flowers, small to larger fruit, an entire plant or leaf, pick it off, and then after it has presumably made the Gus reporter RNA, which is then expressed to become a protein, you bathe that in the substrate, and then that will turn blue. So you can see here everything is blue. That means our assay is working well. And then we take a negative control and we put that in the substrate, and that shouldn't turn blue, and it doesn't. You see that this is all just sort of bleached out. You put it in alcohol so that you get rid of the green, because the green, or the chlorophyll, because that will overlay the blue color, and so you can't see it very well. That's why it's kind of washed out. And then you have the gene of interest. So in this case, this gene is called IMA. It doesn't matter what that gene is. I'm just showing you as an example that you can see here that when you fuse the IMA promoter with the Gus reporter, and you take individual parts from the plant to see where the IMA gene is expressed, you can now see that it's expressed in the fruit, but it seems to be maybe not so much early on, and then later on it seems like it's in the seeds, but not at this stage. But at this stage it is expressed in the seeds, and it's mostly here at the very top of the fruit, not so much in the leaves, not really in young seedlings. So this seems to be a fruit-specific gene. And so using this reporter construct, you can now tell that this gene is expressed at certain developmental stages and not at others. Okay, so that is a Gus reporter. Now let's come back to our discussion of auxin. How do we use a Gus reporter to better understand how auxin is involved in the gravitropic response? So what people did here in this experiment is they used a Gus reporter protein, and they fused that to the promoter of an auxin responsive gene with an auxin responsive promoter. So this entire auxin responsive promoter is now sensing auxin just like it did before when it was actually producing an auxin response protein. But now it's no longer doing that. Now it's fused to the Gus protein. And so whenever auxin is present, and whenever this gene in response to auxin is turned on, it's no longer the gene that is responding to auxin. It's now the Gus gene that is being turned on. It's just being turned on at the time when previously the auxin response gene was turned on. So imagine that as the switch. We talk about the promoter always at this light switch. So the light switch is the one that's toggled on or off, and then whatever it turns on will come on when whatever handles the switch is there to handle the switch. So auxin is flipping the switch, and then this response, this light comes on. And so we have a response to auxin that we can now see because of the Gus fusion protein that is being activated only when there's auxin present. So that's taken this reporter construct idea a step further, right? We're not only interested to see where a certain gene is responsive, but we're taking that so we can also trace certain molecules, in this case auxin, that are binding to those proteins. So this blue light here just shows you the experiment that was done. So a plant that is growing upright is grown, and then it's bathed in the substrate. So this is one of those fusion protein Gus construct plants. And you can see that as long as it's growing upright, there is blue substance down here along the stem. And this blue light here just shows that this plant was grown in blue light, and the light came from the top. So when the plant is growing upright, you can see, this is just a close-up of this part. You can see that this particular gene, this auxin response gene, was expressed pretty much everywhere, but mostly in the middle part here, in the stele of the stem. Also a little bit here to the left, also a little bit here to the right. So it's evenly expressed everywhere. So that means that auxin, which turns on this gene, is found mostly in the stele, but also evenly distributed towards the rest of the tissue. And since we know that auxin is transported polarly from the top to the bottom, but it can also diffuse out, if we just look back here at this model where auxin can go through the cell walls and through the tissue by diffusion, but very slowly. Most of it quickly gets transported through these transporters from the top to the bottom. Right? So this experiment then shows that as long as there's no directional growth initiated by either light or by gravity, auxin is evenly distributed throughout the plant. But when you take the plant and you turn the light on and it comes from the side, so this is directional light now, then the plant starts to grow towards the light. And of course, that is phototropism, right? We talked about phototropism. We talked about the involvement of blue light. We talked about the involvement of the blue light receptors, the specific ones that are initiating the directional growth. Now, the interesting thing in part of this part with respect to our discussion today is that you can see that now auxin response genes are only at the top part of the plant. They're not so much on the bottom part of the plant. And if you zoom in, you can see that this part here is blue. That means that the auxin response gene was expressed over here. And that means that auxin must have been found much more over here than over on the other side. And you can even see that the stele is there's hardly any blue color in here. So all the auxin was moved out, coming from the top, but moved out towards the side. And of course, greater concentrations of auxin there, promoting growth there, will then explain the directional growth of the plant towards the light. So let's remember that the reporter indicates where an auxin responsive gene is expressed. The reporter does not indicate directly where auxin is found, right? This is an indirect way of reporting where auxin must be found. Okay, so now we know a way to see where auxin is transported, where it's found, under certain conditions, under certain environmental conditions. What I want to show you next is how it was found that these particular proteins were expressed where they were expressed. So with that, I want to introduce you to two other techniques, or maybe it's just really one other technique that is nice to know for plant cell biology or cell biology, physiology in general. So here we have a root cap. So this is the end of the root of an Arabidopsis plant. And what was done here is an antibody was used against the OX1 protein. So the OX1 protein, remember over here, that is the OX1 protein. So that's the influx carrier. In order to see where it's localized, what people did is they used an antibody. They cut these sections. So this is sort of like an in situ, except you're not looking for RNA, you're looking for protein. So if you then use an antibody that's labeled here with a fluorescent dye, could have been a radioactive label, I suppose, but it works so nicely with a fluorescent dye. And then you look under the microscope where that dye has bound. You can see that it's at the very top. It's not on the sides. It's not on the sides. You can see this little dark spot here. That is most likely the vacuole. So there's no expression of it in the cytosol for sure, and there's just background really in the rest of the cell. And there's a strong expression towards the top of the cell. So that's called immunolocalization. This kind of technique is called immunolocalization. I sometimes call it in situ westerns because it's in place, right? It's in situ, just like the in situ hybridization that we discussed before. But it's using an antibody just like Western blots do. And so it's not the same as an in situ RNA hybridization, it's a protein hybridization. So an antibody to auxin is linked to GFP and then hybridized to tissue sections. Okay, let's, and then that tells us where OX1, the protein named OX1 is localized. A second set of experiments showed us where this efflux carrier is localized. So people were using a mutant screen, and with that mutant screen they found this particular mutant phenotype, and they called it the pin phenotype because it's sort of like a pin-like structure. You see that it doesn't produce a top, it just produces a little pin, and then there are side shoots that are coming out. So with this PIN1 mutant, when that gene was found, when that was cloned, and then the protein was deduced, the protein structure was deduced from the sequence, it could then be used as a reporter fusion to see where it's expressed. And so in this case, this is a little bit different from the reporter fusion that I discussed with you just a few slides ago. And I want to point out what the difference is. So here, it uses a reporter, same as before. It uses the promoter of interest, same as before. So this is the PIN1 promoter. But in this case, it leaves the PIN gene intact. So that means that the PIN gene is expressed, meaning it makes RNA, and then that is translated into a protein. Now that has tagged on at the very end a GFP protein. And when that gets read in frame, so it has to be designed such that that the end of the PIN gene will still be in frame with the transcriptional translational start of the GFP gene, then that means that basically you can make the PIN gene glow. It sort of has this end, this little bit at the end that produces the GFP gene that is glowing. And so you can then see not only where the gene is expressed, but also where the protein is moved to. So in this case, this was done to look for the expression of the PIN1 gene, and you can see that it also is localized to the low part, the low end of the cell file. Now you may say, how do I know it's the top or the bottom, right? That's something that I really can't see very well either in this particular picture. But I suppose if you look in a little bit more detailed, you can probably, make out the outlines of the cell, and maybe the outline of the cell you don't see it at the top. You have to look at lots of different cells. I guess at this point we have to check from this data and from the previous data, we have to take the author's word for it that this was the lower part of this cell as opposed to the upper part of that cell. So PIN1 GFP localizes to the basal end of the cells. And so that has shown us using mutant screens to find mutants, to find genes, to then identify the proteins that were encoded by these genes, and then looking where these genes are found in the cells to deduce the function of these particular genes. Okay, so PIN1 genes are auxin efflux carriers. Turns out pins are a whole family of genes, and the PIN1 is the one that is using most of that auxin from the top and translocating it to the bottom. So from your book, this particular figure shows that PIN1 directs vertical auxin movement from the shoot to the root. But it turns out that all these other PIN proteins are also translocators, except that they are specialized for lateral or tissue-specific transport. And so what this figure shows you is all these other PIN genes that are known, PIN 1, 2, 3, 4, and 7, and they are found in particular parts of the cell. So if you, for example, look here, the PIN 7 gene, right? That is the one that is responsible for lateral transport here from the center stele where the auxin comes down from the top to be moved to the outside, and here as well. But it also seems to be helping from like a reflux where the auxin gets transported into the other direction. So these are lateral auxin transporters. And then the PIN2 gene appears to be one that is transporting the auxin into the other direction. So auxin comes down from here and then goes back up to the sides, but not very far, only of course as far as where PIN2 proteins are expressed and localized in these very outer cells of the root. So multiple PIN proteins are involved in the translocation laterally or vertically through the plant. Okay, let's come back to the gravitropic response, which was the beginning of the whole question. Right? We saw this gravitropic response. We asked, how does that work? Why does it work? And how is it mediated? And then we talked about auxin and how auxin is transported, how auxin is localized on the cellular level, on the tissue level. And so now we can come back to the original physiological response, and we can say, well, when the plant is growing upright, here's your upright seedling, and you place it horizontally, then auxin is located to is located to one part of the tissue. So in the case of phototropic response, it was away from the light. In the case of the gravitropic response, it's towards gravity. And so you see the little red arrows here showing where auxin is transported towards the bottom of the stem, and then you have increased growth on the bottom, producing directional growth, and the plant can grow up. There's one little wrinkle. It's not really a wrinkle, but it's sometimes a little bit hard to wrap your head around, and that is that the same process, of course, also happens in the roots. The roots also grow down. This is positive gravitropism. This is called negative gravitropism, so away from the gravity vector. The reason why transport of auxin towards the lower part of the root tissues causes downward growth, and I write here in the shoot it increases growth, in the shoot in the root it represses growth, is because auxin has multiple different responses depending on the sensitivity of the tissue to the hormone and also to the concentration of the hormone. So low levels can increase growth, high levels will decrease growth. And then if you have a very sensitive tissue, then that sensitive tissue will have its part where auxin can act as a repressor much sooner than one that is less sensitive. And so that is the reason why auxin can have both effects. It can lead to an increase of growth here and therefore upward growth, and a decrease of growth here on the bottom and therefore downward growth. Okay, so the one thing that we don't know about gravity yet and the gravity response that triggers the relocalization of auxin is due to these starch granules. So when you think back all the way to the first lab that we did, you saw these cross sections of roots, and you saw these purple little circular areas. And we talked about them, and we said that they were amyloplasts. Amyloplasts, here are the amyloplasts. Amyloplasts store the starch, but they also have play a role in the sensing of gravity. So if you can imagine that this plant here is growing upright, and it has these two cells, and it has these amyloplasts. And these little lines down here, they are part of the cytoskeleton. Now, it turns out that starch granules, like when you put this on the side, like a jar of marbles, if you put that on the side, they will all fall over, right? They will respond to gravity. They will now change the way that they pressure on the cytoskeleton, and the cytoskeleton tugs on the endoplasmic reticulum. So it turns out that this change here in the location of the amyloplasts with respect to sedimentation in the cell is the trigger for changes in the endoplasmic reticulum. And the endoplasmic reticulum then is able to relocate these membrane-bound auxin transporters to a different place of the cell. And so now auxin is transported no longer going this way, but now going this way. At least that is the current understanding. There's still some uncertainty about exactly how this happens, but the current theory is that transporters are moved or they are activated. So if you have the lateral transporters, maybe these are the ones that are activated through this tugging on the cytoskeleton and the endoplasmic reticulum. So it's not exactly known yet, but we know a lot more than we did 10 years ago with respect to the to this mechanism of how gravity is sensed and then sets into motion the change in the auxin transport. So let me summarize here what we know about gravity response. So the gravity vector changes, starch granules sink to the bottom of the cell and press on the cytoskeleton. The auxin transporters are relocated within the membrane to the new bottom, and then auxin is distributed to the bottom part of the stem and the root, where the stem then grows upwards and roots grow downwards. And I talked a little bit here about the sensitivity of auxin. So you can read back about that as well. Okay, so auxin is important for tropic responses, right? Phototropic, gravitropic responses. We now know a little bit more about how auxin does that. There's the polar auxin transport pathway, and there are membrane transporters that allow for differences in auxin concentrations from the top to the bottom. And then there is a way for these membrane transporters of auxin to be relocated within the cell wall, the cell membrane, and that can then change the stream of auxin within the tissue. So let me show you another aspect of where auxin is important. And while it's a very different developmental stage, the main principle is very similar in that auxin acts by setting up a gradient of higher to lower concentration between different tissues. So embryogenesis is this event that I have in mind. So embryogenesis also requires tissue-specific auxin transport and differences in auxin concentrations. So just briefly here, before I show you a cartoon of this, this is a developing embryo. So you see here two cells, a basal and an apical cell. So this is right after the cell has cell has divided, and now it's dividing again here, and then now you see four cell stage head. You still see this suspender, as it's called, and this is called the protoderm, multiple cells. Now the cotyledons are starting to be visible. Now you can see a little bit of the embryo here, and it gets a little older, and then you see the cell, the seed coat. This is the seed coat. Here's the little rootlet of the embryo, and here you see the cotyledons. So this is a little bit easier to see. So you start with the embryo sac. We saw this when we looked at through the microscope in the very first lab. You see the antipodes, the polar nuclei, the egg cell, and then the egg cell itself is, of course, what is fusing with the sperm cell. The second sperm cell, you may remember, fuses with the polar nuclei to produce the triploid endosperm. But the diploid embryo is then, so the zygote forms, that is then what is producing, of course, the rest of these tissues. So all of these tissues are different in their auxin concentrations. And what is important, much more so than the name of all these different stages and these different colors and these different parts of these different stages of these different developmental seedlings here, is the fact that those different parts are set up because of an auxin gradient. So you see here that there's auxin is moving into a certain part of the plant through transporters and creates a concentration gradient, a difference in concentration of auxin. And so that means that these parts here will become something, and these parts will become something else. And you can see that from the top cell, you're producing what becomes the cotyledons later on. From the bottom cell, you produce what becomes the suspender, the rootlet later on. And you can see that even within what becomes the cotyledons, there will be concentration maxima and minima of auxin. And auxin is moving in all these different ways throughout this early stage developing embryo. And the way that it does that is through the pin proteins. So pin proteins are required for setting up an auxin gradient within the developing tissue, and that establishes the polarity, and that allows the plant to produce little bumps here that become the primordia for the leaves, for the cotyledons, and so forth. So that is why auxin is important as well. All right, so with the last few minutes, I want to go back on the molecular level and think about how these changes that are auxin sensitive, once you have auxin produced, right? This is this little molecule that I showed you at the beginning, but how does that lead to specific changes on the transcriptional, on the gene level? For a very long time, people were looking for auxin, for the auxin receptor. So what is it that in the cell perceives auxin and then acts as a transcription factor? Is auxin maybe a transcription factor itself? Well, it turns out it is not. But that took a very long time until people found out what molecularly the auxin receptor is. It turns out it's kind of complicated. So let's look at this diagram here, this cartoon. It turns out that there are a number of genes in two classes that are called the AUXIAA genes, and then the second class, the ARF, the auxin response factors. And when an auxin response factor, so there's about 20, 25 auxin response factors in this gene family, and maybe the same number of AUXIAA genes in that family, but they can dimerize. So when they dimerize, auxin response factors act as dimeric transcription factors of auxin-regulated genes. So what I mean with that is that this ARF protein would like to bind with another ARF protein as a dimer, dimeric transcription factor. But it can also bind to this AUXIAA protein. Now, when the AUXIAA protein binds to the ARF proteins, that prevents dimerization of ARF to ARF. Right now, there's a dimer of an ARF to an AUXIAA. But these ARF proteins can only become active when they get this monkey off their back, when they don't have this AUXIAA binding to them. As long as they do, they sit at the promoter level of auxin-regulated genes, and they shut down the transcription. So there's no transcription of auxin-regulated genes. That means the plant basically perceives no auxin. There's no auxin around, so therefore the gene is repressed. That's why I say here, this is the first part of the transcription initiation, the repression state. Now, when you do have auxin in the system, so when auxin becomes available, when it's signaling in the tissue where this status of gene repression is currently active, so when it's currently repressing the gene expression, then things change. That is the activation step. So I have here what we saw before. So this was the repressive stage. This is what I wrote before. Now auxin becomes available. See, this is the little molecule of auxin here. It becomes available. And what it does is it binds. Auxin facilitates the binding of the AUXIAA protein to the SCFTIR protein. So this is the SCFTIR protein. Do you see this, the way of writing these protein complexes? We've seen that before for the jasmonic acid pathway. So in this case, this particular response is mediated by this complex binding to this protein. So the SCFTIR, TIR is one protein, SCF is a protein, ABF is a protein. They all complex together. And them together as a complex can now bind auxin, and auxin fits in right in this little groove here. And when those bind, when auxin glues them together, basically, then it makes it available for binding. This is a stronger binding than the binding between it and the ARF protein. So ARF proteins now become available. They are freed up after the AUXIAAs are mopped up by the SCFTIR and the auxin binding. You see that here. So now they are bound together, leaving the ARF proteins available for dimerization. And only, as I said earlier, only as a dimer, the ARFs, the auxin response factors, can lead to transcriptional activation of these auxin response genes. So now the AUXIAA is gone, ARFs binding with each other, you have active dimer transcription factors. They can bind to the promoter, and they can now turn these genes on that they were previously repressing with the help of the AUXIAA genes. Now, what happens to the AUXIAA genes? The SCFTIR complex is a ubiquitination, causes ubiquitination. It's a ubiquitin ligase. So we talked about that also with respect to the jasmonic acid pathway. So we have ubiquitination, and when you have ubiquitination of a particular protein, that's this little tag here of ubiquitin, multiple ubiquitin units get tagged on, and you may remember that that is the kiss of death, the molecular kiss of death. These proteins are now taken to the proteasome and degraded there. And when they are gone, the ARFs are stable and can continue to activate transcription of auxin-regulated genes. So that means as long as auxin is around, these two are able to bind. These can turn on transcription, and so therefore they're responding to auxin without auxin actually binding to these auxin-regulated genes. They're just making it possible that these transcription factors can bind. Well, and that is the end for today. I will see you in class.